Oh, yeah. Okay, I think you should be able to share now.
Do you want to pull up, Taylor? Do you want me to?
Yeah, I will.
Okay.
Okay, can everybody see it? Okay. So we did first part of the metabolism, which is like what's lactic acid and stuff. So I'll get into that once I figure out how to present. Okay, it's behind you guys.
If you go over view, yeah.
All right. So first off, just like lactate, like what is it? So it's just a natural byproduct of anaerobic glycolysis or homolactic fermentation. They're kind of interchangeable terms just based on like what you read. Lactic acid is converted between lactate and via hydrolysis. So that's just because physiologically our bodies aren't very acidic, right? So the bicarbonate that's naturally found within our blood plasma serves as kind of a buffer. And as you can see down here, the reaction that's kind of takes place from it, the only difference is just that extra hydrogen, which I'll talk a little bit about later. But generally speaking, lactate levels are very low in a normal physiological situation, so about 0.3 to 1.3 millimolars per liter. So just keep that in the back of your head too as we're kind of going throughout this. So cellular respiration and metabolism, like as we've all figured out by now, like it's a huge idea. So I decided just to take a step back for a second and just kind of see where kind of things are with respect to that. So we know that there is a bunch of glucose within our bodies, you know, to use as fuel and as kind of a starting point. So we take that glucose and we go to pyruvate with glycolysis in the cytoplasm and get to pyruvate just by multiple steps. We won't even go over that. But then when we get to the pyruvate, we kind of come to a crossroads, you know, like we can either go to the aerobic pathway or we can go at anaerobic pathway. And you know, of course, like as you can see with the diagram, like we want to go to the aerobic way. That's the most like optimal way to get the most payout. But anaerobic is kind of serves our backup, and we'll see in a little bit here that some of the good actually comes from it. So it's not all bad by any means, but it's just kind of like serves the body to be like the most efficient like as possible. So as you can see, like going the anaerobic way, we take the pyruvate or pyruvic acid, and we go into lactic acid or lactate. Those are kind of interchangeably just with hydrogens where they're at. And then just as we know, like oxygen, you can continue through Krebs and go to oxidative phosphorylation, which is the gold standard. You know, we get 36 ATPs. So we'll keep that in mind. But so this extra hydrogen, so like what's it doing? Like what is the purpose of it and where is it going? Sorry. So we know that NADH plus is required for glycolysis, right? So when we get to the end of glycolysis, we have NADH. So that's another benefit. That's one of the benefits for anaerobic glycolysis, because we can change this NADH back into the NAD plus by doing the homolactic fermentation and getting the lactic acid. We also turn that NADH back into the NAD plus, which can be recycled back through glycolysis, just keeping it as like a well-oiled machine, if you will. So again, like here's like a diagram here. So you can see glycolysis is up at the top, and here's this byproduct of the NADH. So you can see the NAD plus is a natural byproduct of lactate, which actually is a good byproduct for us, and then it can be recycled up back through glycolysis, and you know, depending on if maybe it will go back through the aerobic way going a second time, who knows. So tissues like the heart require a lot of ATP, you know, like it pumps all the time, start to finish, until the day you die. Like, so there's obviously going to be a lot of pyruvate that is involved. And with respect to like, there's going to be oxygen there, so most likely you're going to have a lot of acetyl-CoA. So what the body needs to then figure out how to do is turn these large amounts of NADH into NAD plus, which Again, with the hydrogens, like what the body has decided to adapt to do is take these oxidative phosphorylation shuttles to transport these electrons from the mitochondrial membrane. And this has more to do with like the aerobic way, but the point of this whole slide is that if the rate of glycolysis overwhelms these shuttles and it's not able to move the hydrogens back to optimal, that's kind of where the second backup plan comes from, where the NADH then increases, and then the lactate then saves the day, and then it generates the NAD plus needed for glycolysis to continue. So that's just another application of how kind of like the backup plan anaerobic glycolysis is. So again, like here's another like little graphic that I thought was really helpful. So you can see like glycolysis happening within the cytosol. We can get to pyruvate, or we can go into the mitochondria and do the oxidative or the aerobic glycolysis, or we can stay within the cytosol, do the fermentation, get the lactate, and then the recycling. But then you notice some of this is moving. The lactate is leaving the cell membrane. So like, what's kind of the deal with that? So we know, maybe like you've been working out, or a lot of you guys are athletes, and like you always like talk about, hear people like, oh, like my joints are hurting, like there's lactic acid in them, or like I got buildup. And it's like that's definitely a contributor to like where all the rest of the lactate is going. It is partially a byproduct, but it's also going to different places within the body. So I won't go much into this. I'm a biochem major, so if I could talk for days about this, but here's just like where kind of it goes from there. I kind of like left it. I didn't want to leave anything unsaid. So just know that there's more processes like where it can go, and it can go to the liver to be processed further, and as well as the muscles, which it can accumulate in and cause muscle pain, etc. But there's also ways, as you notice, that ATP is also generated just from here. Just you can see, like I said, not much detail, but it's kind of interesting to see like how actually efficient the body really is. So I just kind of stuck that in there. And then also like some more metabolism of the lactate. About 70% of it is processed by the liver and is removed by the liver. But also places like mitochondria-rich cells such as skeletal and cardiac myocytes and the proximal tubules remove the remainder of the lactate by converting it back to pyruvate, which then can be, used and recycled back through either fermentation again or continue through Krebs and into the mitochondria, et cetera, to get more energy. And I thought it was really interesting. Only about 5% is actually like renally excreted. So, I mean, that just goes to show like how much really is recycling is done within the body. So that's the that. So like now, like what about exercise? Like how does that kind of throw a wrench into things? So obviously, like I talked about, like aerobic, of course, number one at the end of the day, like we absolutely want to go there. But if you're running, a crazy amount, or doing a crazy day at the gym or whatever, obviously like your oxygen levels are going to be depleted within your body. So the backup plan then is going to be to use those anaerobic pathways and to generate energy in that way, because oxygen isn't always going to be available as much as we want it to be. So when we are struggling to find these oxygen levels, we can go through and use these anaerobic pathways, like I said, and get the pyruvate into the lactate. We're going to have a bunch of lactate then at that point, right? So to reduce the massive amount of lactate produced, the pH does drop, and the oxidative phosphorylation is going to counteract with the H+, the hydrogen gradient. And then the oxidative phosphorylation is going to also supply the H+ that's going to be used for hydrogen again, or the NADH and NH+, and all of those recycling kind of points. So.
All right. So then the two things I'm sure you're gonna show you a graph later, but I'm gonna explain the two things on the graph. So when we look at it, you can understand it better. So first is the OBLA, which is the onset of blood lactate accumulation. And pretty much this is when your body starts to get tired because you have so much lactic acid in your blood. And then this is going to be higher than the lactic threshold. So on the graph, this will be farther to the right. This will be higher up. We can go to the next slide.
Oh, sorry. I forgot I was sharing.
And then the lactate threshold is just like before you get a large buildup. So like your body is keeping up with putting the lactic acid back, like converting it back to whatever it needs to go back to. So this is like before you get tired, like you can just like keep going all day at this like general rate. Okay. So then here is a graph that shows the relationship between the blood lactate in millimoles per liter and then your workload. And then we chose to graph, we used the second test subject from our homework. And then you can see, can you guys see my pointer or no, since I'm not screen sharing?
Do you see mine? Yeah, okay.
Okay.
I'll try to follow along, I'm sorry.
All right, so a little bit lower than that. It's like kind of above the 100. It's hard to play, I'm not pointing there. Yeah, there, that is the, this is hard, the lactic threshold. So like before then, it's like the first curve in the line. So like before then, you're not gonna get too tired, like you're doing all right. You can kind of keep doing at that general average, like you're like normal. It's not like too intense for you, like a good intensity, but it's not like horrible. And then like the second point, kind of where those lines cross up there, that's the other one where you like after that, like you're gonna get tired and like you can't keep going forever. Like you're gonna have to shut down sometime or slow it down to get back into the lower range. And then for the statistical analysis, we used a regression analysis, and we found that the p-value was 0.009, which is less than 0.05, so that means that it's significant. We can reject the null hypothesis, so we can support that thus far in our findings that workload and blood lactate are related. And then, so after we learned all this information, who would want to use it or like why? So athletes or pretty much anyone who wants to get better or is helping someone get better. A lot of times this can be used for training, a lot of times with people like long distance runners, because a lot of times after a while, like they're not good with getting oxygen after like a long time. So they have to convert to anaerobic. And then a lot of times like tempo runs or like people who are lifting weights, like after a while, they get like, if you don't like, people who lift a lot realize if you don't like roll out or anything, your muscles get pretty sore. Part of it's from like lifting, part of it's from like the acid building up. So yeah, if you understand information, you can realize you gotta try to push the limits and stuff like that. Okay, and then as our references.
Awesome. Very good, guys. Group 2, are you in the house? Janae?
Wow.
How do I stop sharing? Am I stop sharing? Oh, stop.
Oh,
There
it says I
Never-
can't do it yet. Oh, there you go. Is it on full screen or no?
No.
There we go, okay.
Yep.
Okay, so we also looked into different aspects within the metabolism lab that we did. So the first one we looked into was what are the different fuel sources used during exercises or like exercise and different intensities. So we started with looking into at rest and then intensity. And so before I start on the low intensity though, I just wanted to like touch base a little bit on the fuel we use during rest. And so at rest and during normal activities, fats contribute 80 to 90% of our energy source. So a huge portion is due to fats during rest. And then next is carbohydrates, which they provide another 5 to 18% of our energy. And then lastly, proteins only contribute about 2 to 5% of our energy. In lecture she said that proteins have a lot of other processes that they're used for within the body. So we really just rely on carbohydrates and fats more for the fuel sources we use during exercise. And so now on to the low intensity fuel source. For low intensity, we utilize the oxidative system, and it's also known as oxidative phosphorylation. So interchangeably words there. And it's anaerobic in nature. And this different processes, but I figured I'd just go over it again in a little simpler terms. So the first process is aerobic glycolysis, and this occurs in 2 different steps. And the first step occurs in the cytosol, and it involves the conversion of glucose to pyruvate, and then the resultant product turns into NADH. And then this process alone only produces about 2 molecules of ATP. And then the second stage of this process of the cycle, and this is where glucose, fatty acids, and then certain amino acids are oxidized. And then this oxidization of the molecules then are primarily used to transform into the energy that's contained, or the energy that's in these molecules is turned into ATP, so our energy source. And then lastly, the third process is the electron transport chain. And this is a series of 4 different protein complexes that to create an electromechanical gradient that helps lead to the production of ATP within the system. And so some benefits for the oxidative system is that it is very highly fatigue resistant, so you can do it for long periods of time without feeling too much fatigue. And then it also yields the greatest amount of ATP as well. I know I said in aerobic glycolysis it only yields about 2 molecules of ATP, but within the 3 different processes, they yield the greatest amount of ATP within the other 3. And then a con for this, though, is that it is pretty slow to be activated. So next is moderate intensity, and the fuel source to use for that is the glycolytic system, and this is also known as anaerobic glycolysis as well. And the glycolytic system is kind of like the next in line tool after the ATP-PCr system runs its course, which I will talk about next. And this is where carbohydrates supply glucose that circulate within the blood, or is also stored in the muscle and the liver. And blood glucose and the stored glycogen is then broken down to create ATP through the process of glycolysis. And this produces a taxing waste product called lactic acid or lactate, like Taylor and Jess had previously talked about. And this is kind of like the in-between system, so it's moderately fatigue resistant as well. And it also requires 10 to 12 enzymatic reactions for the system to work. So you the difference between aerobic glycolysis, which I just talked about for lower intensity, and then the moderate intensity, which is the glycolytic system. And so finally, we have high intensity, and the fuel source we use for that is the ATP-PCr system. And this system consists of adenosine triphosphate, which is the ATP in the name, and then phosphocreatine, which is the PCR. And this energy system provides immediate energy toward high energy phosphates. So in the picture, actually, I think this might be, if not the same one, a very close one to the one that was in lecture. And so you can see that ATP is here, and through the use of ATPase, this bond is broken down. One phosphate is broken off, so then it makes it into adenosine diphosphate. And the breaking of that bond causes the energy, or ATP. And then same thing down here, the breakdown of that causes the ATP, or the energy, to be released there. This fast system, it only lasts 3 to 12 seconds, so very short-lived in nature. And then some benefits include it's a super fast system, and it is also a very powerful system, way more powerful than the other 2 systems I previously talked about. However, a pretty big con for this one is it's not very fatigue resistant at all, so fatigue sets in super fast.
All right, so comparing the lactate threshold versus the ventilatory threshold. So the lactate threshold is when the lactate production becomes greater than the lactate clearance, causing buildup of lactate in the blood. And then the ventilatory threshold is determined from ventilatory changes that reflect trends, changing your CO2 extraction, your O2 consumption, and breathing volume and rates. It's compared to the lactate threshold, where we collect blood samples at progressively higher intensity levels. So these both are set up to find the trend change towards accelerated or accumulating fatigue problems, and they both occur at similar levels of intensity. So the higher the threshold gets, the harder it is to come back down to normal state, which was mentioned in the level is at 87%.
Okay, so then the second question we looked into was how are the metabolism and the pulmonary or respiratory system interrelated? And they work together, and so they're directly correlated. So in order to carry out metabolic processes within the body, we of course need oxygen for those cells to be able to work. And then the pulmonary system works to ensure that oxygen is found throughout the entire body. And then it also helps to remove those waste products that are made throughout metabolic processes. So kind of an example of showing just how integrated they are is that, so respiration is increased as cells burn more nutrients for energy during like higher intensity workouts, or so oxygen is necessary, which means then more carbon dioxide is produced as a byproduct. And that carbon dioxide then needs to be released from the body somehow. And that is how the pulmonary system comes into play and just shows how really integrated they are. And then just in simple terms, like metabolism produces and uses energy made within the body. And then the pulmonary carries out that byproducts and the metabolic waste throughout the body and then into the environment. So in this visual, you can kind of see oxygen comes in, CO2 is released as a byproduct, goes through the circulation of the body, through the respiratory or the pulmonary system, and then is released back out into the environment after.
Okay, so looking at the class data from the lactate lab, it showed that as Mary and Brian's workload increased, their heart rate and their RPE increased as well. So therefore, the faster the heart rate and the RPE, the more lactate that will be produced. So for example, looking closer at Brian's workload, it started off heavier as he approached 9 minutes. His workload was at a 210, and that increased his RPE and his heart rate. And that caused him to produce 12.4 millimoles of lactate. So on the next slide, kind of looks deeper in. So we chose to do our statistical analysis over Brian's data. Just a term to go over before I talk about this slide. As Jess mentioned earlier, the OBLA stands for onset of blood lactate accumulation, and it is at the point during exercise where lactic acid builds up in your blood and fatigue sets in. So it's at a point where you essentially just hit a wall, but it's also known as the lactate threshold. So for Brian, it shows when he gets to 9 minutes is when he hits his lactate threshold at 12.4 millimoles per liter. So it shows that in our data at the green line, when it starts to rise up, that's when his lactate starts to increase.
Thank you, ladies. One question. This can be for anyone. So again, you talked about lactate threshold and ventilatory threshold and how they're going to occur at the same time. Does one cause the other one?
I think the threshold builds up to a certain point, because like you said in lecture, it's different for everybody, like what their like certain point is, like, but you said arbitrarily, like the 4 millimolar, like is kind of like the point, I guess, like where they meet, and then it turns into the obula. But like you said that it can also like shift and change, you know, per person. But I think like the threshold causes the obula, right? Like it, over time, it accumulates.
So which one happens? I know they occur simultaneously, but when we think about ventilatory threshold, are you trying to get more oxygen in, or are you trying to get more waste products out? What's the goal of more ventilation or more breathing?
From what I took from it is that you're trying to get more oxygen in.
So think about what's happening after the lactate threshold. What happens on that right side of the curve? What type of metabolism are we relying on? So again, once I get to that right side, that's high intensity. So what type of fuel or what type of energy systems am I using then?
Anaerobic?
Right. And so with anaerobic, do I care about oxygen? I mean, I always care about oxygen, but not really, right? I'm trying to get waste out, okay? So again, when we look at the relationship between lactate threshold and ventilatory threshold, the reason that ventilation is increasing is we're trying to get those waste products out, okay? We're already maxed out at that point on oxygen consumption. We're already like to our limit. So the only thing we can do now is continue to have metabolism occur via those anaerobic pathways. So the goal is to get the waste products out. So again, I don't know, you could probably say that lactate threshold causes ventilatory threshold. I would say that's fair, just because as we increase intensity, we're trying to get more waste out, essentially. And that's what's going on with the ventilatory threshold. Okay, neat. Last group, Val. Val, we can't hear you. Try hitting your volume up button. Nope. Try and mute yourself and then come back. Nope.
Now?
Yes.
Yeah.
Okay. Okay. The mic was off.
Okay.
Let me pull this up here. All right, so today, here, I'll start presenting. Sorry, it's like loading, it won't go to full screen. Okay. Is it okay if I just present like this? It's not popping up to full screen.
Yeah.
Or, okay. Okay, well, there it goes. All right, so today our group will be talking about metabolism and endurance performance. So first off, I will just start off talking about the mock data that we saw in class. And the graph isn't. So for, I'm just going to pull up an Excel worksheet so we can see all the data points of Brian. So we chose our subject, Brian, for the data points. And looking at the graph, we can see the lactate threshold, or OBLA, which is used interchangeably with lactate threshold.
Val, if you all we can see is your PowerPoint first slide right now. So you might need to do a new share maybe.
Okay. All right. Can you guys see this now?
Yep.
Okay. Sorry about that. So first off, just viewing the graph, we will see that his lactate threshold is about at around 4.3, and this is also used interchangeably with the OBLA, or the onset of blood lactate accumulation. So what the lactate threshold is, it's like a spike in our blood lactate levels. And then what the onset of blood accumulation is, it's like the set point of our blood lactate. So, or when our blood lactate levels reach about 4 millimoles per liter. And then I'll go back. And then we see that the R coefficient is at 0.835. Is at 0.835. So therefore we can say that that the blood lactate levels did increase and change as the workload went on. And then I'll just go back to the presentation. And then next, Elisa will be talking about how we decided to use heart rate and workload for a predictor for endurance performance.
Are you playing something?
Yes.
We can't hear or see. So when you go to share screen, I think there should be an option that you can like share computer audio or something.
Okay.
We're gonna get this figured out.
Okay. Let's see here. There. Share screen, and then
At the bottom, I think the bottom like left, maybe there's like a spot where you can check share
okay.
computer audio, maybe.
All right, so let's just try this again.
A big portion of endurance, or even another way that we thought endurance performance could be tested was by heart rate. The reason for this is because cardiovascular is such a big portion of endurance, or even activity or exercise in that matter. I actually remember a doctor asking me when I was very young that if I was an endurance runner, I have never, or nor will I ever be a distance runner in my life. But the reason for that is because my heart rate was very slow. Normal non-athletes' resting heart rates are between 60 and 100 beats per minute. And well-trained athletes' normal resting heart rate is closer to like 40 beats per minute. That's a little bit lower on the lower end, but it can get down to that. The lower the heart rate, the more efficient the heart is.
And then next I'll just be relating this to the lactic acid threshold as well.
Another
It was working fine yesterday. I don't know why it's.
That's okay. Just you can just share this with me afterwards, and I'll go back and post it for everybody to hear.
Okay. Okay. So after that, we'll hear Jelani talk about a scholarly article that we found in relation to endurance performance and heart rate.
In the article, prediction of performance by heart rate derived parameters in recreational runners, it described to the researchers that heart rate derived parameters are predictors of endurance performance and are used to monitor training intensity. Also, heart rate at a given intensity or an intensity at a given heart rate are related to endurance capacity. Though heart rate monitoring is not acceptable or not the greatest measurement, it has a major advantage. This advantage being that it's easily measured due to people and athletes currently wearing monitors such as Apple Watch and heart monitors. In the study, the researchers had 130 recreational male athletes who were free of cardiovascular, musculoskeletal, and metabolic diseases. They performed a cross-sectional study where the athletes completed increments running tests until exhaustion. During these running tests and in between, heart rate was monitored, and at the end of each stage, lactate concentrations were measured. The runners completed a maximum running test at 1% of the slope on the treadmill, which started at 9 kilometers per hour without a warm-up. Throughout this test, the speed was increased by 1.5 kilometers per hour every 4 minutes until hit about 14 kilometers per hour. Immediately after each stage, blood samples were obtained and the earlobe and lactate contractions were measured by portable lactate analyzer. So after the study was done, our understanding of heart rate decreases during exercise at a given intensity as the training status improves is accepted because during the exercise at the same heart rate intensity increases. Our results also demonstrated that the maximum heart rate, if it reached submaximum running speed, it was better predictor for the running performance in these athletes. Also, in relation to our mock data for this lab, our research and article proves that because of the workload is increasing, the blood lactate levels are increasing along with the heart rate. And our article would show that the blood lactate levels increase as well after we've seen the lactate threshold. Lastly, some limitation of our study the article that we researched was that it was all males, and therefore they're unable to make conclusions as to how female runners' results will be. Also, that the athletes that were chosen were based off the performance from the previous year, so therefore their results could be unexpected due to performance of the year that the study was done.
And here are our citations, and thank you for listening.
Okay. Thanks, everyone. Okay. So other things I want to do today. I want to quickly go over the neural lab report. I got all those, or your neural lab homework. I got all those graded. You should have received feedback from me yesterday through the eLearn. So check that, because it takes some time for me to go through and give you guys feedback. So please look at those. I want to go over a few things with that, just because I think it'll be helpful for you as you are working on your lab reports. So let me share my screen. In particular, I want to talk a little bit about question number 1 and then the caffeine question. Okay. So question number 1 asked you to choose a disease, describe its physiological effect, and then summarize 3 different journal articles. Okay. We're doing a really good job of finding the key takeaways from your journal articles, but when you summarize a journal article, I want you to give me more than that, okay? So let's say you found something that, I don't know, caffeine is an effective treatment for people with migraine headaches, okay? And so a lot of you, pretty much all of you, did a really nice job of being able to say, this is what the study found. These 2 authors, they found that if you consume caffeine, then it'll reduce your symptoms of migraine headaches. And that's great. I want you to say that, but I also want to know how they know that. Okay, so what was the research done? So they took, what was the purpose of their study? So they wanted to, Smith and Jones wanted to determine whether consuming 100 milligrams of caffeine each day would decrease symptoms of migraines. They took 150 subjects who regularly experienced migraines. They had them take the caffeine, every day for 3 weeks. They measured how many times they had symptoms of migraines. They found that the individuals that consumed the caffeine had significantly less symptoms than those who didn't. Okay, so again, give me all that information leading up to your kind of takeaway message, so that we can then also, as readers, make our own decisions. You know what I mean? So I want you to try to start to think critically. Don't just take their word for it, oh, this guy said caffeine is going to help migraine headaches. I want you to critically think about it and look at some of those details, so that we can then kind of make a decision for ourselves on that. Okay. APA formatting, again, if you didn't do that correctly, I gave you some feedback there. Some of you were spot on. Some of you need a little bit of work with that. There's an example lab report posted for you on eLearn under the cardiovascular section, and you can use that as kind of a guide if you want to, in terms of how to do that APA formatting. Just a few notes on the caffeine question as well. Statistical analysis should be a repeated measures ANOVA. Okay, so we had 1 group and multiple time points. Okay, so more than 3 time points. So you had baseline, 30 minutes, 60 minutes, 90 minutes. So you had 4 time points, but 1 group. Okay, so repeated measures ANOVA was the appropriate method for that. Again, when you write scientifically, don't use I, we, my, things like that. So we want to write in 3rd person. And then some of you just found 3 scholarly articles about caffeine. The idea here is that they should support or negate your findings. So if you found that heart rate wasn't affected by caffeine, okay, you need to find articles that are related to that topic. Okay, not just that caffeine is going to help people with Alzheimer's or caffeine enhances endurance performance. Well, that's great. I'm happy that you're learning those things, but again, we want to apply this to what we're actually doing. Okay, so if you used, heart rate as a measure, and you want to look at that endurance study or whatever, find a way to connect those things. Okay, and same thing when you're using these articles as support, you got to give the details. So the purpose of the study was to determine if, heart rate was affected by caffeine supplementation. They found that heart rate was modified significantly and we didn't. And here's why, right? So you have to be able to kind of figure out a reason, why did they see significance and we didn't? And some of you guys did an awesome job with that, saying, okay, well, we only had a few subjects. Or maybe you want to look at the dosage, right? How much caffeine did our subjects have versus theirs? Were there different forms of caffeine? So a pill versus coffee or something along those lines, maybe that affected it. So being able to kind of draw those kind of critical thinking points, I think will really be helpful for you guys when we move to the lab reports next. Any questions on that? Okay. So that is that. I gotta mark that off my list. Okay, so I think what I will do next is go over the cardiovascular lab, which you have for today. Let's see here. Two-part lab. But you could do it all in one day. How many of you have experience with taking blood pressure? 2? Okay. This is like a skill that I'm like, this was like almost a hard stopping point for me for like this class, because like I want to be there to make sure you guys know how to do this skill. I have posted 2 different little videos for you on how to do blood pressure. Again, just practice is kind of, I guess, the best thing I can tell you for this. So you're going to, for the first part of this lab, basically you'll get blood pressure at least 3 times in a seated position, and then you'll also have them lie on their back. Okay, so they'll lie in a supine position, have them lie for a couple minutes prior to getting those measurements, and then also in a standing position for 2 minutes as well. Okay, resting pulse rate, I want you to actually get like a manual pulse. Okay, so again, go on like the radial side of their hand, use these 2 fingers, don't use your thumb. Okay, and you're going to get their pulse on that, on the thumb side there. Okay. Again, why do I want you to get a manual pulse? Because it's a skill that is important for you to have if you ever needed to. I know that there's all sorts of different heart rate monitors and things like that, but this is a skill that you'll need to know. What if the technology fails? What if you don't have a heart rate monitor and you need to get that heart rate? I want you to have that skill. Okay, so do a manual pulse for that first part of the lab. Okay. That's pretty much it for the first section. Then the second part of this lab, you're going to, I'm going to do a new share. Ask somebody to do some exercise for you, okay? So again, get their resting levels. If you already did that for the first portion, maybe you're using the same person, then you don't need to re-get their resting levels if you do all this today. Okay, so have them do 5 minutes of low intensity something, then get their blood pressure and heart rate, then have them do something more moderate for 5 minutes, get their heart rate and blood pressure again, and then 5 more minutes at more of a high intensity and get their heart rate and blood pressure then, okay? Ideally, like in real life, if we were in class, we would do this on a bike so that you could leave that blood pressure cuff on and get it right away. I understand that we don't all have stationary bikes at our houses. So do the best you can if they could walk and then jog or something like that, so that you can kind of mimic those intensities. That would be lovely. Okay. And then the other thing that you'll do, all these calculations you'll be able to figure out, those are fine, is for resistance training, you'll have them do a wall sit. I was really like ambitious when I first started teaching. I thought everyone could do 5 minutes of wall sits. Turns out they can't. So have someone do, if they can do a minute, that would be great. And when they think they're almost done, then get their blood pressure at the end of that minute. Again, I posted a video demo of me doing this with my husband, so you can kind of see how to do that blood pressure while they're doing the wall sit. Okay. But if, you know, just communicate with them. If they feel like, oh, I'm almost done, you know, like get the blood pressure then at the very end of that exercise, so that we can get the best results that we can. Okay. Again, you can use your heart rate monitors for the aerobic and resistance portion of this. Okay. And then use your blood pressure cuff for that portion. Questions on the activities of.
Is this second part of the lab due tomorrow in the morning as well?
No, none of this is due until Friday.
Okay.
At 8 p.m.
Okay.
Or wait, you have to post it so that the class has data? Is that how I had it set up?
I don't know, but I won't be able to have it done by tomorrow morning because there's something due tomorrow morning. I didn't know if that was this because I don't have anyone at home to test.
Okay. I bet that I set it up that way so people would have the data ahead of time. I'll take a look at it tomorrow morning, and if there's not enough data posted, then I'll post some previous data from previous years as well.
Okay.
I understand you have to like convince somebody to like.
Because I don't have anybody at home because he's gone at work and I work till 10. So.
Gotcha. Yeah. Yeah. Just do it when you can and post your results when you can. And like I said, I'll check tomorrow morning and post additional data if we need it for the class.
Okay.
Okay, let's see. Any questions on the blood pressure stuff? Okay. EKG, again, I believe I talk about this in the lecture as well, which you'll have today. But you're basically going to go through and label these components of an EKG and label the conduction system, and know the relationship between those two things, because that'll be on probably a quiz or an exam for sure. That's the lab activities. Okay, I am going to go over the lab reports. Again, I would encourage you to look at the example one that I posted. It's not the same topic, but it'll give you an idea of how to format things. Okay, so again, in your introduction, define systolic blood pressure. What is diastolic blood pressure? Why are these important components for us to measure? Again, your intro shouldn't be more than 1-2 pages max. Okay, please double space. Well, I guess it doesn't really matter because I'm going to be responding via technology. I guess I don't care if it's double spaced. Normally I like write in between the lines and stuff, but anyway, do whatever you want. It doesn't need to be more than 2 pages though. Okay. So just, define blood pressure. These are important variables because high blood pressure is a predictor for cardiovascular disease, blah, blah, whatever. Okay. Make sure you cite your sources. The purpose of our experiments. Okay. So the purposes of our experiments in the first section were to practice the skill of measuring blood pressure. The purpose of the aerobic exercise bout is to determine the change in blood pressure with aerobic exercise. The purpose of the wall sit is to determine the effect of resistance exercise on blood pressure or something like that. Hypothesis of our experiments. Okay, so again, what do you expect to happen with the systolic pressure? What do you expect to happen with diastolic pressure? Okay, so you should really have 2 or 3 purpose statements, or purposes, I guess, for the lab, and then 2 or 3 hypotheses as well. Okay. Again, use third person. Don't say my hypothesis is blah blah. It's the hypothesis of the current investigation or something along those lines. Okay. Methods. So again, you want to describe what you did in enough detail that someone else could come in and do the same experiment. Okay. So pretty detailed here. And then in your results, you're going to do statistical analysis on the class data that you have. What else? You can include a chart or a graph of some sort if you want. Just make sure you label your axes there. Again, I think the example one that I gave, she's got a chart for each like different variable or experiment that she did, okay? Okay, so in your results section, you just simply say, this is what we found. That's all you have to do. In the discussion, that's where you get to talk about why you found what you found, okay? So the majority of your time should be spent in the discussion section. How did we compare to what we hypothesized? How did your individual subjects' results line up with what was expected? Any sources of error within the experiment, okay? Don't say there were no errors. There's lots of errors. There's always errors when we're using human subjects and human researchers, okay? So some examples of potential error. Can you think of some? Anybody? Can you think of any error?
Whether or not like something's calibrated or not can introduce some error, or like say I measure somebody's pulse radially and say somebody else does it maybe like different way that produces different results or something.
Sure. Other potential errors. I'm not asking you to like have your subject not consume caffeine, okay? They could be on some medications or caffeine that might affect blood pressure, heart rate variables. So again, we're not controlling for any of those factors. Some of you might use your mom who is not fit or active at all. Some of you might use a roommate who is fit and active, right? So our subject population will be very diverse, I imagine. Okay. Again, when you think about discrepancies or errors within the experiment, give me like a paragraph. It doesn't need to be like 19 pages. Some people spend like 4 pages on errors. It's 1 point. Just give me a short, give me a short paragraph that lists a couple different potential options. Okay. The majority of your time, again, you want to think about, you can use your lecture notes for this section. Why does systolic blood pressure change? Why does diastolic blood pressure change? How is it during aerobic exercise, and how is it with resistance exercise? Okay. And then lastly, your directive section, again, summarize and discuss the effectiveness of one scholarly article. So again, think about those details that we talked about with the neural lab. Okay, so what was the purpose of the study? What was the subject population? How did they set it up? What variables did they measure? Statistically, what did they find? And then what was the takeaway? Okay, so make sure you give those details there. Okay, questions on the lab report setup? Again, these are pretty time consuming. Once you do one though, they're not as bad, I guess. I think that's true from what I have heard around the world here. Okay. You also have a pulmonary lab, which is tomorrow. I don't think there's anything crazy that I need to go over with that. You have like your pulse oximeters, and there's a brown paper bag in your equipment stuff. I think all that's self-explanatory, but I'll answer any questions on that on Friday. Let's focus on getting your cardiovascular stuff going, and then the pulmonary stuff will be tomorrow. Okay, so I'm not going to go over that one. Okay, questions on anything coming up, any of those assignments? Okay, so the last thing yeah, go ahead, Val.
So for the cardiovascular lecture, like I was typing notes about it yesterday, and at like about slide 23, it started to like speed up, and like it just kept going like from slide to slide to slide.
Huh.
And yeah.
Okay.
So I don't know.
I'll look into it. That's weird. Like I was just talking faster or?
No, like there's like no sound, but I could just see like it going from side to side to side. And it was like 40 minutes in length. I don't know.
Okay.
That was the
I'll
time.
look into it. Maybe when I uploaded it, something went wrong. So I'll go back. You said slide 23. Okay. I'll look into that after class today. Other questions on stuff? Anybody? Okay, so the only thing else I want to do today is spend just a little bit of time reviewing those energy systems, okay? So if one of you wants to get the whiteboard going and share your screen, let's start with the ATP phosphocreatine system. I just want to make sure everybody feels good about this. I feel confident you guys have the lactic acid, you got that system covered. I think you did a nice job of explaining that with your presentations, but let's go back and take a look at ATP phosphocreatine here quickly.
Do I need to share my screen?
Yeah.
Okay. Find it again here. Whiteboard. All right.
So take me through this system, the ATP phosphocreatine system. What do you know about it? How's it work?
So that we need like the ATPase and the creatine kinase. Or just shooting out ideas, or I'm sorry.
Yeah, no, you do it. You go, girl.
Oh, okay. So it's the first step is ATP and the ATPase split into the phosphates, which we get the energy from. Oh, ATPase, which is hydrolysis, so we get the H2O there. And then the second one.
Wait, hold on. I need to write this down. What do I start with?
So we have the ATP, and then the ATPase is the enzyme that comes in and breaks off one of the phosphate groups.
ATP what?
So it has yeah.
Okay.
And then the ATPase comes in and snips off one of the phosphates, and then there's water produced in the hydrolysis.
There, he's cutting it.
And then you can do equal sign, and then ADP and energy.
And what?
And energy, like yeah, cool. And then we break we have P and the creatine, and then we change, or the phosphocreatine, is that the term for it? And then we break the phosphate and the creatine apart, and then yeah. And then we can do like plus energy there too. That's step 2.
This is going to be our little energy symbol.
And then step 3 is the phosphate from step 2 comes in and regenerates the ATP to form step 1. So it's like recycles.
It goes to the phosphate goes to the ADP.
Yeah, and then it comes up. Yeah.
Perfect. The only thing I would change is that when the phosphocreatine breaks off, we don't actually get energy. It just like fuels, like we take that phosphate molecule and we can yeah, we'll get energy eventually because then it hooks on to that ADP. But yeah, that's just delightful. That's exactly what I wanted to see. Where in the cell does this occur?
The cell cytoplasm.
Yep. And is it a high power or low power system?
High power.
High power. And is it really quickly fatigued or not very quickly fatigued?
Quickly fatigued.
Yep. And how long could it last?
3 to 12 seconds.
Very good. Okay. What about lipolysis? That was in your metabolism lecture, yeah? Let's do a new screen. Let's see what's going on with that. And then I'll let you guys go for the day.
How do I do a new screen? I forgot. Wait.
I don't know.
I think you can scroll down, can't you?
Clear. Wait, is this gonna work? Clear. There we go.
Okay.
Which one are we doing now?
Lipolysis.
How do I spell this?
Lipo.
Lysis.
Yep. Yeah, buddy.
Okay. So we need a fat molecule for this. And then it's like a,
The thing on the outside?
I think, yeah. And then it needs to go to like the blood tissue and the muscles.
How am I supposed to draw that?
What are we looking at now?
This is the fat cell, the circle one.
What's on the outside of it?
I don't know. Part of the cell.
Draw like a little river beside it for like a blood vessel, I guess. I don't know.
On the outside?
Well, I would do like get rid of your outer layer of your, get rid of your outer circle. Yeah. And now do like a little river beside the fat.
Like all around it, just like on one side.
Just like a one-way river.
Like surrounding the whole thing like this.
No, just like just the one side. Yeah. Like the river's running beside the cell. Yeah.
Okay. Here's our little river.
Yeah. Perfect.
So do we need the breakdown of the triglyceride? Okay. And then that will release the free fatty acid.
So we're breaking this down.
Yeah, so maybe do like the 3 tails, like the glycerol and then like 3 tails.
That's just so confusing.
Okay, so let's imagine that the circle here is a cell. Okay, so inside that cell, you're going to store a bunch of triglycerides. So there's a bunch of these molecules inside.
Okay.
So a triglyceride kind of looks like an E, exactly. Yep. So your straight part is the glycerol backbone, and then each branch of the E is a fatty acid. Okay. Now what?
And then once those free fatty acids break off, they go into the bloodstream.
Yep. Yep.
So, and then they enter the skeletal muscle, and then they gain like 2 ATP. And then like this converts them into like acetyl-CoA this is after they reach the mitochondria. Sorry, Jess, I'm doing this like really out of order.
So they get the ATP and then they go to the mitochondria.
Yeah.
And then they turn into acetyl-CoA.
Yeah. They go, yeah. And then, so then this serves as like, as a gateway for sugars and fats to get through the molecule.
And so acetyl-CoA enters into which energy pathway or which energy system?
Carbs.
Yep, Krebs. So the only thing to note here is that this ATP that Val was talking about, we have to invest 2 ATP. We don't gain ATP at that point. So it like costs us money basically to move those fatty acids into the mitochondria.
Is that enzyme then working within the fat cell, and then is it breaking it down and then moving it to the skeletal muscle? Is that what's going on?
Say that again, Taylor.
Is the hormone sensitive lipase breaking the free fatty acid into parts and then putting it into the skeletal muscle?
So the hormone sensitive lipase will be like at the level of the fat cell.
Okay.
Yep. So if I had a lot of hormone sensitive lipase floating around in my blood, I would be a good fat burner. Yeah, that helps us to mobilize stuff out of storage, basically. Good. Questions on this? You feel like you guys have a good feel on the energy systems? Yeah? Okay. Good. All right. I think that is everything I wanted to cover today. Again, on Friday, my plan is to answer, it'll be a short day on Friday. So answer any questions that we might have on the pulmonary lab. And then I'm going to go over the exercise physiology and environmental labs just in case I have these babies early. But I think I'm going to make it to next week. I think it's going to be fine. Everything's fine. Although I had a doctor's I know. I had a doctor's appointment yesterday, and I'm like 3 centimeters dilated and 70% now effaced, which like, that's fine. I like barely progressed this week. I'm like, so I can run now, right? And she's like, no. I'm like, but like a little bit, can I run? No. Like, well, like they could make it and not be in the NICU, right? No. She's like, just settle down. I'm like, I don't know if I can. I'm just gonna see. Let's just see what happens. So we'll see what happens. I don't know. I'm not guaranteeing that I'm not gonna run, but I think we can make it till next week. I feel good about it. Not that I'm in control of anything, but that's my story. So I will plan to see you on Friday. I feel good about that. And yeah, let me know if you have questions. Otherwise, that's all I got today. All right. Val, I'll check into that lecture issue and then I'll let you know. Maybe I need to read it or something. So, all right. Have a good day, everybody.
Thanks, you too.
Am I staying after?
What's that?
Am I staying after to go over the